Hi, good morning all. Today we have to discuss about the topic C4 pathway, C4 pathway or H and slack pathway, H and slack pathway, H and slack pathway, C4 pathway or C4 cycle or H and slack pathway. It is one of the most important topic in the chapter photosynthesis. You have already studied about the topics light reaction or photophosphorylation and dark reaction or C3 cycle or Melvin-Calvin cycle. Now we have to discuss about the topic C4 pathway. C4 pathway is also known as H and slack pathway because this cycle or this pathway is described by H and slack in the year 1966. This H and slack pathway is also known as C4 pathway because the first stable compound formed in this cycle or pathway is 4-carbon compound oxaloacetic acid. Okay, why this cycle is known as C4 pathway? Because the first stable compound formed during this process or this cycle is 4-carbon compound oxaloacetic acid, and this is a peculiar type of cycle which is present in plants adapted to dry tropical region. The plants in the dry tropical regions possess a particular type of cycle in addition to C3 cycle or dark reaction or Melvin-Calvin cycle or biosynthetic phase. This cycle is known as C4 cycle. Why these plants possess C4 cycle in addition to C3 cycle? Because they are present in the plants in dry tropical region. As an adaptation to overcome these conditions, these difficult conditions, they possess a special type of anatomy in its leaf. That anatomy is known as Kranz anatomy. We have already discussed about the topic anatomy, stem anatomy, anatomy of fruit, anatomy of leaf. Okay, in anatomy of leaf, there are 3 types of tissue system are present in the leaf anatomy. Which are they? The epidermal tissue system, the fundamental tissue system and the vascular tissue system. Epidermal tissue system with the epidermis, stomata, cuticle, hairs, etc. and leaf hairs are absent. And the fundamental tissue system in the leaf anatomy is known as mesophyll tissues. Mesophyll tissue is differentiated into 2 types of cells, palisade parenchyma and spongy parenchyma. Okay, but in the mesophyll tissue is differentiated into palisade and spongy parenchyma cells in dicot plant, but in monocot plant the mesophyll cell is not differentiated. Okay, the third tissue system is present in the leaf anatomy is the conductive tissue system or vascular tissue system, which possesses vascular bundles, xylem and phloem for the purpose of conduction of water and minerals. Okay, this is the vascular tissue system present in leaf. Here xylem and phloem is present and these vascular bundles are surrounded by a bundle sheath cells. Here the plants in the dry tropical region or they possess Kranz anatomy. The vascular bundles are specialized. Vascular bundles are made up of large cells and they are multi-layered or several layers are present in around the vascular bundles, and they have many chloroplasts and thick walls which are impervious to gas exchange, and no intercellular spaces are present in between these vascular bundle sheath cells. This type of specialized bundle sheath cells, wreath-like bundle cells, are present in these plants, these C4 plants, and this anatomy is known as Kranz anatomy. Okay, what is the peculiarity of C4 plants? They possess a special type of anatomy, and this anatomy is known as Kranz anatomy, vascular bundles are surrounded by a bundle sheath cells. These are specialized, these are multi-layered without any intercellular spaces and arranged like a wreath. Hence, this anatomy is known as Kranz anatomy. Clear? Okay. Which plants are the examples of C4 plants which shows Kranz anatomy? There is a code to teach it well. Which are the plants which shows the Kranz anatomy? There is a code for it, MASPE, and also another code, SMS AMAR. MASPE means M for maize, A for amaranthus, S for sugarcane, and again another S for sorghum. And this PE stands for pennisetum, means the bajra. Which are the plants that shows Kranz anatomy, or the plants which are known as C4 plants? Example for the plants which are known as C4 plants are maize, amaranthus, sorghum, sugarcane, and pennisetum, means the common name of pennisetum is bajra. Okay, another code for these examples of Kranz anatomy, the plant shows Kranz anatomy, are SMS AMAR. S for sugarcane, M for maize, another S for sorghum, AMAR for amaranthus. Here this pennisetum or bajra is not mentioned here, so both these codes are very helpful to study the examples of C4 plants. Clear? Okay, now we have to discuss about the C4 cycle, the cycle present in the C4 plants in addition to C3 cycle. The C4 plants, C4 cycle takes place in two cells, the mesophyll cells and the bundle sheath cells. Okay, now we have to discuss about the C4 cycle. There are two types of cells are involved in this cycle, mesophyll cells and bundle sheath cells. So for my Malayali students, there is a code to recognize or recall the C4 cycle. Mesophyll cell is a mandabuddhi, MB for mandabuddhi. Okay, M for mesophyll cells, B for bundle sheath cells. Okay, clear? Okay. Now we have to discuss about the C4 cycle. It is a cyclic process. First, the carbon dioxide from the atmosphere or released from the mitochondria is accepted by a 3-carbon compound PEP, phosphoenol pyruvic acid. So phosphoenol pyruvic acid, a 3-carbon compound, which is the carbon dioxide acceptor in this C4 cycle. Okay, this 3-carbon PEP is bind with 1-carbon compound or carbon dioxide to form a 4-carbon compound, oxaloacetic acid. Oxaloacetic acid is the first stable compound present during C4 cycle or formed during C4 cycle. Hence this cycle is known as C4 cycle. This oxaloacetic acid is transported to bundle sheath cells and now it converted to another compound, 4-carbon compound, which is known as malic acid. Okay, here NADPH is converted into NADP plus. Okay, this malic acid malic acid is a 4-carbon compound. From the malic acid, 1 carbon dioxide is removed. This removal of carbon dioxide is known as decarboxylation and it is converted into 3-carbon compound pyruvic acid. This the carbon dioxide which is released through the process of decarboxylation is enters into the Melvin-Calvin cycle and sugar is formed. In C4 plants, Melvin-Calvin cycle, C3 cycle is also present in the bundle sheath cells. The remaining 3-carbon compound pyruvic acid is transported back to mesophyll cells and here the phosphorylation process, the removal of from the phosphorylation process, that is ATP. From ATP, one phosphate group is released and it is converted into ADP, and this phosphate is added to the pyruvic acid and phosphoenol pyruvic acid is formed. Hence the process is begin from the PEP and end in phosphoenol pyruvic acid. Hence it is a cyclic process and it is known as C4 cycle. Clear? Thank you.